Lecture 2, Blood and Lymphatic System. So in this lecture, we are going to be looking at the various functions and components of blood, as well as the genetics of blood. For example, if you're type A blood and your spouse is type B blood, what's the probability that your child is going to have type AB blood? Right? That kind of genetic problems. We're going to be looking at some of those. Then we're going to look at the lymphatic system, which is actually a connection between the blood and the immune system that we are going to be looking at. So let's get started. Blood. There are approximately 5 liters of blood in an average person. That's 70 kilogram. Depending on the weight of the person, depending on the gender and various other factors, this number might be a little bit higher or maybe a little bit lower. But you know, on average, we're talking about 5 liters of blood for someone that's 70 kilogram heavy. What is blood? Blood can be described as a protein-rich fluid with cellular components. Okay, so the protein-rich fluid is mostly water, and that portion of the blood is what we call plasma. Right? So the plasma makes up about 55% of your total blood volume. So if you take blood from a person and then you spin it in a centrifuge is basically a machine that rotates at really high speed. So by spinning the blood at really high speed, you are going to be able to separate its content based on its mass. So the heavier stuff, like the cells, are going to sink to the bottom, and lighter stuff, the liquid portion, is going to float to the top. So this top clear yellowish layer is the plasma, and that makes up 55% of your whole blood. The cellular components are called formed elements. So these formed elements include things like RBC, red blood cell, white blood cells, as well as platelets, and we're going to look into these cells in more details in the upcoming slides. But they are the formed elements, and they make up 45% of your total blood volume. So if you look at the spin down blood, the formed elements are going to be at the bottom. The red cells are going to be heaviest because they contain iron in the hemoglobin. That's what allows them to transport oxygen, and that iron makes them a little bit heavier, and so they sink to the bottom. In between the plasma and the red cells, you are going to find a cloudy layer that's just kind of hovering in between. We call that the buffy coating. That's where you will find the white cells and the platelets. So here is an actual picture of blood that has been separated by centrifuge. And again, here you have your 55% plasma, and here you have your 45% formed elements, rather. Now, the plasma, as I said, is the fluid portion of the blood, and it's mostly water. Right? Later on, we'll talk more about blood transfusion, but when we donate blood, your blood is actually separated into plasma and into red cells. We cannot take the whole blood from one person and transfuse it into another person. Okay, that will almost always be harmful, right? So we're going to look more into why that is the case later on. But here is an example of frozen plasma. They can actually freeze this stuff and then thaw it when you need to transfuse it into someone. So in that plasma, mostly water, but you would have things like dissolved ions. So these ions are what we call like electrolytes, right? There are sodium ions, for example, potassium, chlorine, chloride rather, so on and so forth. And all these things are going to be dissolved within your plasma. There are of course nutrients, right? Nutrients like glucose, right? There might be some amino acids, and there are going to be waste products as well. So waste products include things like carbon dioxide, right? A little bit of urea, which is from muscle metabolism, right? And there are going to be a lot of proteins. Okay, remember the plasma is the protein-rich fluid, right, off the blood. There are thousands and thousands of different types of proteins dissolved in your plasma. And you might remember from last lecture, we talked about osmotic pressure, right? So part of the osmotic pressure is created by these proteins, right? They help to retain the fluid inside your blood vessels. So functions of blood, well, we have three different types of formed elements. These are the cellular component of your blood. They make up 55% of your total blood volume. So of that, a majority of it would be the red blood cell, okay, RBCs. They are also called the hematocrit. The main function of the hematocrit, the red blood cell portion of the blood, is to deliver oxygen. Right. So in the red cells, there are these hemoglobin. You might remember learning it last semester. It's a protein with 4 subunits in its quaternary structures, and it's going to be carrying oxygen and delivering it throughout the body. More on that later. We have white blood cells. White blood cells help defend the body against pathogens. Pathogens are basically anything that any living thing that can make you sick. So, for example, bacteria is an example of pathogen, virus, fungus. So, all these things, if they get into the body, we want to have a way to fight them, to deal with them, and that's why we need the white cells. And last but not least, we have the platelets. Platelets help stop the bleeding if you have a cut. So let's talk more about the proteins that are found in your blood plasma. Like I said, there are thousands of them. We're just going to look at a few examples. The most abundant protein in your plasma, the one that you have the most of, is something called albumin. So albumin is made in the liver. Your liver is on the right side of the body, just underneath your diaphragm. There's your diaphragm, right? So right there, that's your liver. Liver actually makes a lot of the plasma protein, and one of them is the albumin. You would find albumin in things like milk powder, in egg whites, right? So a lot of albumins in our food sources, but we make our own albumin as well. And the major function of albumin is basically to maintain blood osmotic pressure. Right? You remember the capillary exchange discussion we had. Right? We have a lot of things dissolved in our blood vessels, and if that concentration is higher than the surrounding area, then that creates a gradient and osmotic pressure that will draw fluids into the blood vessels. The proteins, for the most part, reside within the blood vessels because they're too big to leave the capillaries, right? And that's what's creating the condition for you to retain the fluid, to draw fluids from the surrounding tissues into your blood vessels. So if you are having protein, if you're malnutrition, rather, sorry, like kids in developing countries, sometimes, if you think of like a World Vision poster and showing for kids who are malnutritioned, they are skinny everywhere except for the belly. And why is that the case? Well, if you are malnutritioned, then you have low proteins in your diets, probably, right? And if you don't have enough proteins in your diets, then you don't have the raw material. You don't have the amino acids that are required to make the albumin. And if your albumin level is low in the blood, then some of these dots kind of start to go away, right? And now there is not as much of a difference in terms of solid concentration between the blood and the outside, and so you're not able to bring back as much fluid from the surrounding area, and that leads to edema. That causes the fluids to accumulate in your belly, for example. Now, if you move your arms around or if you walk around, then the muscle movement will actually help recirculate some of those fluids back into your system, but the abdomen, we don't really move that too much, and as a result the fluid will accumulate and resulting in the swelling of the belly if you are malnutritioned. There are various other plasma proteins. Many of them are important for immune responses. That's to help fight the pathogens, for example. And then there are some that are responsible for coagulation. Right? Coagulation is basically to stop bleeding. Right? If you have a cut, you're going to need some of these proteins to help you stop the bleeding. So we're gonna explain this in more detail later on. It's kind of like an overview, foreshadowing kind of slide. For coagulation, for blood clotting, we have something called fibrinogen, and we have something called prothrombin. Right? So these are going to be circulating in your plasma, and we're gonna need them if you have to stop your bleeding. And we have a lot of these things called antibodies. Your body can make antibodies to fight for your immune defense purpose, and a lot of these antibodies are going to be circulating in your plasma. So where do the blood cells come from? They actually all came from the same place, which is your bone marrow. So in your bone marrow, so if you think about like a typical image of a bone, a long bone, right? In the middle of it, it's a hollow space, and that is where your bone marrow is. Okay, so there are red bone marrows, which is where new blood cells are going to be coming from. As you age, the red bone marrow gets converted into yellow bone marrow, and they're mostly for fat storage. So within the red bone marrow, you are going to have some multipotent stem cells. So stem cells are basically these cells that can become different cell types under the right stimulation, and you have some of those in your red bone marrow. So the process of the stem cells becoming red cell, white cells, or platelet, that is called hematopoiesis. Okay, hematopoiesis. So this big word basically means blood cell production. Okay, so each of these paths are going to be a possible destination for the stem cell, right? Under different chemical signals, under different types of stimulation, then the stem cells can commit to one of these pathways and become, say, a red cell or a platelet, or one of these 7 different types of white blood cells. Okay, so they all are going to undergo hematopoiesis, and once they commit to a particular lineage, they have to go all the way through to the end, and there's no going back. Meaning, if a stem cell is going to become the red cell here, they cannot go back and then change to a platelets later on. Right? It's a committed pathway. Okay, let's look more into the cellular components of blood. Here you can see a blood spear. You have the red cells here, and then you have a white cell here. These are a little bit bigger, and they tend to have multiple nuclei in some cases. Let's start with red blood cells. They are the most abundant blood cells. And remember, when you spin down your blood, right, you will have your RBC all sink to the bottom. That portion of the blood is called the hematocrit. Okay. So another name for red blood cell is erythrocytes. Okay, erythro means red in Latin, and cyte is cell. So these are red cells. So some people get confused between what's the difference between erythrocytes and hematocrit. The hematocrit is basically referring to the portion, the red blood cell portion, when you separate the blood. Okay, and whereas erythrocytes, it's literally another word for red blood cells. So here you can see a lot of red cells, and they are kind of like a jelly-filled donut that has been pinched in the center. We can see that there's the indentation right in the middle there. So some of the characteristics of red blood cell is that they do not have a nucleus, right? It's because they don't have a nucleus, that's why they can have this indentation in the middle, right? There's no nucleus in the way that that's going to be taking up too much space. So without a nucleus, like I said, you can have that biconcave shape. So if you look at a red cell from the side, right, that's what it's going to look like. This is concave. This is concave. Concave means it's kind of caved in, right? So it's concaved on both sides. That's what we call biconcave shaped. And the reason for that is you will be able to have more surface area for gas exchange, right? The more surface area you have, the more efficient you will be when it comes to diffusion. Inside the red cell, you will have something called hemoglobin. This is a hemoglobin protein, and in the middle of the hemoglobin protein, you have something called the heme group, right? The heme group. That's the iron-containing component of the molecule, and this heme group allows you to allows the molecule to bind to the oxygen and deliver it to different parts of your body. If you have a deficiency or defect in hemoglobin or RBC, then you might have anemia. So in sickle cell anemia, for example, there is a mutation in one of the nucleotides in your DNA sequence. So normally it's supposed to be T and A, and this codon would code for glutamic acid, but in a mutation that becomes a different amino acid, and as a result of that, your cells are going to look different. It's going to have this moon crescent sickle cell shape, and they're not very good at delivering oxygen, and so the person is going to become anemic. So anemia in general just means you're not going to be able to deliver oxygen effectively, so you're going to experience things like shortness of breath, and you might be a little bit tired because you're not delivering oxygen to the body as much as you need to. So this is just one type of anemia. Like I said, it's not always about the hemoglobin problem. You can have an RBC problem, right? If you are suffering from excessive bleeding due to injuries, for example, then you might have hemorrhagic anemia, right? That's anemia caused by excessive blood loss. So whether it's a problem with hemoglobin or RBC, if there is a deficiency or a defect in them, then you're going to have some form of anemia. So just a little bit of genetics here. So normal individuals, they are going to be, say, big S, okay? This is going to be the normal phenotype, normal genotype for someone who does not have anemia. And because the mutation is a single base substitution, in order to get the disease, you would have a small s, okay? And that would means you have sickle cell anemia. Okay, so of course, if I ask you, which one is better, the answer is going to be the normal phenotype. You're not going to have the symptoms of anemia. However, people who are sickle cell anemic, they actually are immune to malaria. So malaria is a disease that is spread by mosquito. You might have heard of that before. And it turns out if the person is sickle cell anemic, because of the shape of the red cell, the parasite cannot go into these cells to replicate. So the person is not affected by malaria. But if you have to choose between malaria or sickle cell anemia, that's a difficult choice. It's neither of the options are very good, right? And then it turns out there's a third option. Right? If someone is big S, small s, so these people are going to be carrier of the sickle cell anemia gene, but they're not going to exhibit the full symptoms of sickle cell anemia. So these people are going to be relatively normal, and they are going to be immune to malaria at the same time. So if you take a look at a map here, where there is prevalence of malaria, shown here by the green, those areas tend to have a high frequency of the big S, small s, that is the heterozygous genotype, because those people with this genotype are going to be protective against the malaria. So that's just kind of an interesting thing to see, the correlation between the genotype of someone and its correlation with a particular disease such as malaria. So, as mentioned earlier, the RBC, the red cells, are formed from the red bone marrow stem cells. In fact, all blood cells come from these bone marrow stem cells, but here we're talking about red cells. They too come from the red bone marrow. And it turns out what happens is if there is a decline in RBC level, your kidney will release a hormone called erythropoietin, or EPO for short, which will then stimulate red cell production. Okay, so if you have low RBC, right, then that's going to cause your kidneys to release EPO. Okay, and then EPO is going to stimulate your bone marrow to make more RBC. So this is what we call like a negative feedback, right? So the trigger is going to cause a series of events that would result in the opposite event taking place. So you have low RBC, then the hormone is going to cause you to have high RBC. Okay, so that's an important pathway to maintain homeostasis. So some athletes, for example, they would train at higher altitude, and with the thinner air, then your body is actually going to secrete the EPO to help produce more RBC, so that you're going to be able to meet the oxygen demand. And so when you come back to sea level, now you have more RBC than before, then you might be able to deliver oxygen even more efficiently than you were before. So EPO can actually be used for cancer patients who are anemic due to the chemotherapy or radiation therapy. A lot of times the chemotherapy and radiation therapy have significant side effects because they target rapidly dividing cells, right? And examples of rapidly dividing cells would be cells in the bone marrow, because we keep on making new blood cells, the patient might become a little bit anemic if they are undergoing through these treatments. So to counteract that, you will inject them with EPO. So we can make artificial EPO through biotechnologies, and that can be given to cancer patients. Also, EPO can be given to people who undergo dialysis treatment. Right? In dialysis treatment, for people with kidney failure, for example, a lot of times the blood cells die as a result of going through the tubings and the machines, and they too can become a little bit anemic. So with the injection of EPO, that could adjust for the side effects. So a typical red cell is going to have a lifespan of about 120 days. So that's roughly 3 to 4 months. And when the red cells are too old, they will be trapped in the liver or the spleen, where they will be eaten up by a type of white cells called macrophage. We're going to look more into macrophage later. So if you're not sure, if this is the person, this is the diaphragm. Your liver is going to be on the right side, and your spleen is actually going to be on the left side, just behind the stomach. We will learn more about these things later. If you're not sure where they are, you can just Google them on your own for now. When you break down the red blood cell, you want to recover as much of the components as possible, so we can recycle them and use it for production of another red cells, for example. So when you break down a red cell, the hemoglobin is going to be released from it, and we will recover some of the protein components from the hemoglobin, right? Maybe break it down into amino acids that we can use to build something else later on, and we want to recover the iron from the hemoglobin, and then the rest of it are going to be metabolized. It's a long process, but what I want you to know is eventually you will break it down into something called bilirubin. Okay, so the hemoglobin, whatever you can take back, you take back. Okay, and the rest of stuff is going to be metabolized, broken down into something called bilirubin. Now, bilirubin is going to have a yellowish color, and typically we remove them through urine and feces. So That's the reason why they're the color that they are. Is because of the bilirubin content. So later on in the lecture, we're going to talk about how some babies are born with a very high level of bilirubin because in part due to blood incompatibility with the mother, and that could result in the baby looking a little bit yellowish. So you might have heard of a condition called jaundice. Right, so more discussion on that later on. So that's it for red cell for now.